We have started gene therapy in our earlier class, and we have seen some of the things into it. What is gene therapy, or what is somatic gene therapy rather, and what are the things which are related to it. Let us go ahead. So we have found that in gene therapy what we are doing is we are trying to replace the faulty gene with the corrected gene, and to transfer this correct gene we are using one or other kind of techniques or vector which carries that, or delivery system or such disease which will make it. So we have got various conditions that we have seen, either we need it for chronic conditions or we need it for acute conditions, and we have seen it the difference between chronic condition and acute conditions. Now again this also we have seen that what are the considerations for the DNA transfer, that in that we have seen that what is the molecular size of various classical synthetic drugs, what are the molecular weight or size of protein drugs, what will be the molecular weight of nucleic acids, and accordingly what can be the situation, how it can be delivered, whether it can be given orally or only parenterally. These are various things which we have discussed. These are the different situations where their pharmacokinetic behaviors will be different, and all this we have studied in a different way. Then next in this is what we have discussed earlier. There are three classes of gene delivery, and either it can be ex vivo, it can be topical, it can be systematic delivery. This is also we have seen. Then next in this, what we have seen here is two classes of gene transfer vectors. These are viral, non-viral, and we have seen the issues which are specific in that. We were almost in the middle of it. What are the differences between viral and non-viral vectors are there? Vectors, I told you, these are the modalities or the way how we are transferring or we are carrying the gene to the exact place where we need it. So viral vector, when we say that these are having packaging capacity from 4 to 30 kilobytes. Kilo means 1000. So 4000 to 30,000 base pairs we can put it, and that is okay. The gene we required is a small, but if we require bigger one, example in case of dystrophin gene or this cystic fibrosis, this receptor gene is there, that is CFTR. In that cases, these are bigger than 30,000 base pairs. Then in that case, it will be problem. Here it is not an issue. Very large, like up to 150 kilobytes, kilobase pairs, you can put it into normal. Then toxic level ratio, that is infectious and non-infectious particles from 1 as to 10 or 1 as to 100, it is there. Here a very small percentage of non-relevant this material is there, so again non-viral vectors are that way good. Then strong immunicity, they are either present in the capsid and envelope, proteins are there, residual viral genes are there, so they have strong immunogenicity they can show because of these different kind of proteins which are envelope proteins and viral genes. It can bring out very strong immune reactions which can be prominent. While the non-viral vectors can be moderate immunogenicity, some of these are methylated status of DNA, and in that cases it can be strongly immunogenic. Then next one in that, in the contaminants are there, the replication competent viruses are there, or some viruses which are wild type of viruses are there. If they are contaminated, they can cause very big havoc because it can result into that viral infection. Here it can be the pathogens free from poor DNA purification, and some endotoxins can be there which are because of non what we can say because of difficulty in purification of the non-viral vectors. The viral amount obtainable with recombinants is very poor. If it is with the recombinants, while the amount of DNA molecule is usually not a problem, but the other component depends on the chemical synthesis. The complexity of production is always there. Existence or not of packaging, it can be there. Whether this type of DNA means first we have to insert this DNA into viruses, and then we have to see that it is at proper place if they are attached. While in that it is not a such big problem in non-viral. So this problem has to be solved. For that, each has got their own advantage and disadvantage, and accordingly we can select the vector as present. So ideal properties, what from all these different we can what it should be stable. Wherever whatsoever vector is there, it should be making a stable one. Particle should resist the serum inactivation or enzyme inactivation, and particles should be inert to the immune inactivation. The particles should bear a tissue blocking specificity DNA. Construct should be include the tissue specific regulation there. Efficiency, it should be able to deliver it whatsoever DNA is there at particular site and do it activation. Cargo means the DNA load which is there, it should contain nuclear translation signals, it should include the genome integration functions, and accordingly it should get integrated and it should get expressed. Then other particles should not include immunogenic or toxic actions. Then it should not encode immunogenic or toxic actions in that. And next in that is it should include anti-apoptotic functions. It should not cause apoptosis. And then next features in the weight is, if we compare what are there, what are the small parade of various popular vectors and methods. So one is viral and another one is non-viral. Viral is example are adenovirus, adeno-associated viruses or retroviruses including HIV. While those which are non-viral vectors, example of these are naked DNA. Naked DNA means without any coat. In viruses there is a coat. So naked means without any coat, it is open DNA. Then next is the liposome, etc. These are various formulation in which DNA is incorporated, and oligonucleotides. These are various formulation techniques. Everyone is having its own positive and good things, so nobody is perfect. Something is good with one while something is good with the other, and some are the problem with one and some are the problem with the other. Let us see what are these. So first one is recombinant adenoviruses. Recombinant means where the adenoviruses DNA are combined with another DNA, we call them as a recombinant adenoviruses. So there are various approaches into it. Generation 1 approach, generation 3 approach, then hybrid adenomas, and then this adeno retroviruses, adeno-associated viruses, adenotransposons. These are various ways by which we can use adenoviruses. What are advantages? It can give capacity up to 8 kilobase pair in generation 1, and in more than 30,000 base pairs capacity in generation 3. Adeno can be grown at very high titers. They can be very fast. Only what are the problems? They do not integrate. It can contain some problems. They can be toxic. And the example of this which we have tried in the various deficiencies in cystic fibrosis and in oncolytic viruses, in that it was tried. Then next is recombinant adenoviruses or adeno-associated viruses. So what were the approaches? Like helper-dependent production, helper-independent production, then cis-complementary vector and co-infection. These were various techniques which were used, which were planned. Now you can find the deficiency wise good, but specificity wise and toxicity wise it was not acceptable. Here same thing, it is specificity and persistence wise, means it cannot be repeated again and again. efficiency, maybe it is allowing good amount of base pairs, but the problem was there and toxicity was significant. So here it is, the next one, this adeno-associated viruses. Advantages are persistent in the genome permits long, so it will remain there. It is helpful for the long-term expression. High titers are easily obtained. Immunogenicity is very low, so that is good sign. Only problems are what? The small capacity. It can only go less than 4.5 thousand base pairs, which do not allow to accommodate important genes or gene clusters. There are many such are there which are bigger one, and therefore they are not possible. They can be useful only for very small kind of insertion of DNA. Then recombinant retroviruses. Uh, Here we can have variety of such viruses. Murine means various rat or mice related species. Then VSV serotype viruses, lentiviruses, self-inactivating combination viruses. So these are different viruses. Here again, efficiency wise good, persistence wise good, but problem is specificity and toxicity. So it can got capacity up to 9,000 base pairs, and it can integrate through the transposition is also in a quiescent cells. The quiescent cells means which are not uh, doing any activity, those which are in resting one. It permitting principal long-term treatments, but only problems are insertional mutagenesis. So before that, if it is getting mutagenated, then it can make problem. Then it can cause gene silencing. That it can stop some of the genes to express. So it's stopping gene to express means gene silencing. It has got very high mutation rate, which are very dangerous, and low titer are there. So in certain cases, example in Parkinsonism and in adenosine deaminase deficiency, it was used. Then naked or complex DNA. These are the direct DNA, no coating, no insertion in viruses or like that. So example are they are available as a naked DNA injection, naked DNA with pressure jets, naked DNA with electroporation, liposomal formulation, or combination of any of these are various formulation techniques. Naked DNA direct injected to the site is a direct injection. Naked DNA plus pressure jet like things are there which will be pressured at certain place. And naked DNA with electroporation means application of charged lipid formulations are there. The problem with these are many, uh, they can carry unlimited size. They have comparatively lower immunogenicity than viral one. They have lower biorisk means infection risk of non-viral formulation is, but problem is very low efficiency of gene transfer and even lower stable integrations. So these were tried for critical bleeding, ischemia, cardiac ischemia, vaccination, and anti-stenosis, particularly when stents are put and stenosis occurs. In that it was tried, and these were the problems which were there. Then oligonucleotides, they can be added as antisense oligonucleotide. It can be ribozymes, it can be DNA zymes, it can be added as a triple helix or decoy computation or gene correcting oligonucleosides. So these are various oligonucleotides are there. Advantages are there. They are suitable for handling dominant defects, transient treatments, that is gene modulation, or permanent treatment. Example of anti-cancer treatment, restenosis, muscular dystrophy treatment in various models, and it was found effective in animal studies. So this is all about various gene therapies which are present, various vectors which are there. Example like adenoviruses, retroviruses, then other naked DNA and in other solutions. Then we call them as a what problem is there is antibody response, there is a limiting packaging are the problems. Then solutions are there using of synthetic viruses, or they are called as a virosomes. Synthetic viruses means people, as I told you earlier also, people are feeling or thinking that Even this COVID is a synthetic virus, means it is synthesized in laboratory. So similarly, synthesis viruses are there, they can be there. Then biologic bombardment of local direct injections are there, or electroporation, liposome of this direct or naked DNA. We can have these various techniques. Liposomes, gene correction are there, but they are not very efficient. They are inefficient in transport. Because of the low transfer efficiency, very little impact is there. So all money, all investment can go waste, and therefore most probably people will go for these synthetic viruses or virosomes, which are artificial, which can do or which can maybe having still better capacity to carry the gene load. So these are the problem. Not all gene therapy approaches are random shooting. Many efforts are made at various places for various diseases. Example of that, this it can be genotoxic or it can be non-genotoxic. So these random integrating vectors, example of these are lentiviruses, retroviruses, then adeno-associated viruses, plasmids are there, then plasmid plus transposons are there, and they are tried. Then transient but which are not integrating vectors are like adenosine, adenoviruses, plasmids, RNA virus, then oligonucleotides, then artificial chromosomes, and there are some specifically integrative vectors like hybrid vectors, base 31 integrase base, designer integers. Gene correction vectors are there like geminoplast and single stranded DNA. So these are all variety of ways which can be either genotoxic, which cannot be genotoxic, and they can be used in various treatments. So now if we just make all the sum total of it, then we can realize what are there. So which vector will be important for which disease. We have seen what are specific things, particularly important things were four. That is whether these are capable to carry the bigger gene molecule, what is the capacity to carry, whether they are able to insert it properly, whether they are safely admin or reaching to the place without getting destroyed, and what kind of the toxic or studies toxicity effects they are having. And one more we can have that what can be the repeatability and what kind of period it will be taken. So in various cases, what are so far studies are saying, example of chronic metabolic diseases like Gaucher diseases, hemophilia, then hematopoietic diseases. So these are various viruses like adeno-associated lenti, adeno3, retroviruses. They are most suitable because persistence of expression they can able to make, and therefore minimize re-administration because it is very costly procedure. Then local chronic or progressive, example CNS joints, etc., where the problems are there. Here again adeno-associated virus, non-viral like plasmid or naked DNA lentiviruses. Here no rapid expression is necessary, persistence is required, and low toxicity is required. So these are helpful. Solid tumors and metastasis, example of cervical, breast, brain cancers. So adeno2, plasmids, oncolytic recombinant viruses can be there. And why they are selected or why these are suitable, because they are rapid, they are having transient expression of cytotoxic or immunomodulators. In case of traumas or infection, example trauma because of infection, fracture, burn, wound, acute infection, anaphylactic, etc. In that case again adeno2, plasmid, then modulatory oligonucleotides, they are useful because we want rapid and transient action which is required into it. So these are there. There are some technologies which are not exactly gene therapy, but yes, they do as well similarly, or their function is in that way. Example is bioactive oligonucleotide like antisense technology, decoy dsDNA, double-stranded DNA, decoy RNA, ribozymes, DNA zymes, then siRNA. These are various bioactive oligonucleotides. They can be synthetically made and they can be administered. Then other are like oncolytic viruses like Onyx 15, Onyx 68. Then R, that is R means recombinant, wherever R word is there. So R adeno means recombinant, or HSV, we shall see what recombinant is. Then implants of encapsulated cells can be there. So these are places where so far they were tried, and their results were there, they are published. Example of this ADA deficiency or immunodeficiency, cystic fibrosis, hemophilia B, then SCID is an immunodeficiency disease, limb ischemia, cardiac ischemia. In various such conditions, various genes are transferred, and these genes, what were the results into that, there are some publications which shows impact or effect of it. There are many studies were conducted, but so far not big results are there. The success is limited, and therefore the studies are there. They are now reaching towards a peak. Uh, in last few years, it started somewhere in 1990, and they are now picking up. So these are various studies are going on at various places. Major areas of frustration means, although everything was found good, but still it was big failure is there. Example was muscular dystrophy. Uh, that was 1 in 3,000 newborn male was having muscular dystrophy, and it required persistent expression, extremely large gene. It takes around 14,000 base pairs, and 2 mega base pair gene is there. It is not clear whether the regulation is necessary. It is not clear at which point disease is irreversible, because initially it is reversible and then it becomes. Then another is cystic fibrosis. This, incidence is 1 in 2,500 newborns. Luminal attempts, they are failed because anatomical biochemical barrier, no receptors, muscle layers, large in are there, that requires probably regulation. It requires long-term regulation. It is unclear at which point the disease become irreversible. So these are various there. All these genes were discovered in early 90s. No suitable vector was there, no satisfactory delivery method was there, and therefore there is a failure. Most feared potential side effects of gene transfer is immune response to vector is there. I told you it can be a very severe immunological reaction. Immune response to new or foreign gene product, it can be there. General toxicity of viral vector, it can be there. Then adventitious contaminants in the recombinant viruses are there. Then many times it is causing risk of cancer because of the insertional or random integration in the genome. That mutagenesis will cause cancer risk. Then next is contamination of germ cell lines, and they are still related to the primitive state of the this viral delivery. So there are various experiments which were beaten. I will not go for more that detail. We have seen what is gene expression, gene therapy, and how this gene therapy can be effective, how this gene therapy can make the treatment, this one. Now the last topic which is remained is the recombinant technology. So let us see about this, what recombinant technology is. So recombinant technology is the DNA molecules, they are formed in the laboratory. Means they are not natural ones. They are made outside that. We call it as a molecular cloning. That means duplication, whatsoever it was there, and to bring together the genetic material from multiple sources, creating sequence that will not otherwise be found in the genome. So this is a new one is there. This is a general name for the piece of DNA. that has been created by combining at least two fragments from different sources. This recombinant DNA is possible because DNA molecules from all organisms share the same chemical structure and they differ only in nucleotide sequence within the identical overall structure. So this advantage is taken. If it would have been different, if suppose human beings are having ATGC, adenine, thymine, guanosine, and this cytosine, instead of that, it could have been a different nucleotide with instead of pyrimidine or pyrimidine or purine, if it is different heterocyclic compound, then it would not have been possible. Then it would have been totally mismatched. But fortunately, nature has provided all people has got DNA having same nucleotides. So because these are there, same gene sequences are there, then it can be inserted into one or other, and that is insertion from one to other is called as a recombinant technology. So they are they are similar in chemical structure, they differ only in the nucleotide sequence. The recombinant DNA molecules are sometimes also called as a chimeric one. Okay, because they can be made of material from two different species, and therefore they are called as a chimeric. Since they are coming from, chimera is a mythological character. It was a monster which was fire-breathing hybrid creature of the there, and it was having just like Narsinya we are having. Okay, in that means half is a man and half is a lion, or Ganpati ji we can have some portion elephant and some, so we call it as a chimera. So this is there, we are having chimeric DNA, that is two DNA are different from different sources and they are combined, so we call it as a chimeric DNA. This recombinant DNA technology uses palindromic sequences and it leads to production of sticky and blunt ends. So palindromic sequences, these are the nucleic acid sequences in a double stranded DNA, or they are in RNA, wherein the reading in certain direction of one strand matches the sequence reading in the same direction on the complementary strand. So if you remember, अगर WhatsApp किसी को याद है तो उसको palindrome बोलते हैं, you will be knowing. The words are there, come this the words are there which are from both side, both direction, from this side to that side, they are coming same. That is called as a palindrome. So they are having palindrome techniques and they leads to production of sticky and blunt ends, which is one end is sticky and one is blunt, and by that way they are making these various DNA sequences. Now the DNA sequences that are used in construction of recombinant can originate from any species. There are many examples actually we are using nowadays. Those who remember diabetes, we have seen that nowadays we have got recombinant human insulin. Earlier, when we were studying, only there were two types of insulin were present, that is oxine or bovine insulin and the porcine insulin. No human was possible because these insulins were obtained after slaughtering of the animals and extraction of insulin from their pancreas. But after availability of recombinant technology, now we are, what is done is this the DNA which is coding insulin, human insulin, is put into certain microorganisms. It is attached, and because of this attachment of this DNA, now this bacteria will able to produce human insulin as a byproduct during their metabolism. So this is how this biotechnology or this recombinant technology is helping. So this recombinant technology, what we are doing is, it can be from any species. Example, as I told you, human this coding for insulin is put into the bacteria and they are producing it now. So using DNA technology and synthetic DNA, literally any DNA sequence may be created and it is introduced into any of the very wide range of the living organism. That can danger also, but that has resulted into many benefits. Proteins can result from expression of recombinant DNA within living cell. They are called as a recombinant protein, or we have seen R word, either recombinant viruses, recombinant proteins, recombinant DNAs, recombinant RNAs, it can be there. And when recombinant DNA encoding a protein is introduced to a host organism, It is not necessarily produced. Expression of foreign proteins requires the use of specialized expression vectors and often necessitates significant restructuring by foreign coding sequences. So it differs from genetic recombination that in the former, in the DNA, recombinant DNA it results from artificial methods in the test tube, while genetic recombinant is a normal biological process that results in the remix of the DNA sequences in all organisms. So like that, DNA is cloned, that is copy of important portion of the DNA is made. So first part will be DNA creation. The molecular cloning of the DNA will be done. We know how it has done earlier, that is some of the portion is there, it will be copied, and then it is most important methods are there using polymerase chain reaction. So this is method which is very widely used to make the million of or billions of the copies of the specific DNA sample. Even today we are having RT-PCR test which is very popular for COVID testing, you must be knowing about it. So this polymerase chain reaction, that means there are many enzymes which are there, and one after another they will be adding nucleotide into that. So this addition of nucleotide will make whole DNA, so they can make artificial DNA. It is used to direct replication of any specific DNA sequence chosen by the experimentalist. If experimentalists want that it should be AG, 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 so it will be AG, 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 they can put into, and then it can insert, and now a new DNA which was not existing in that particular species, which is having AG, AG only, it will be inserted. So this is how this experimentalist can make it. So there are two fundamental differences between this method. One is molecular cloning involves replication of DNA within a level cell, while the PCR do it outside the cell. And it is the cloning involves the cutting and pasting DNA sequences, but PCR causes amplification by copying an existing sequence. So in cloning we cut it and paste it. We make suppose three introns are there, we combine together and make a combination of it and make it separate one copy of it. Here it is not like that. It is, if you know that this is a sequence, you have identified this is a sequence, then you make it like AGT or AGU, whatsoever it may be, or AGC. Okay. So go on making AGCA, 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 whatsoever number you want. This can be synthetically produced. Formation of recombinant DNA requires cloning vector. The DNA molecule that replicates within a living cell vectors are, we have seen vectors just now in gene therapy. They are generally derived from plasmids or viruses, and they represent the relatively small segments of DNA, and they are useful in the transportation of the DNAs. In standard cloning protocols, the cloning of any DNA fragment essentially involves 7 steps. One is choice of host organism and cloning vector. DNA therapy we have seen just now, gene therapy. Similarly, at what cell you want to put it and who should be carrying. If it is in naked DNA, then injection or various formulation. Then preparation of vector DNA. Then if we are having vector, then we have to prepare vector DNA so that that initial DNA of vector will able to go for attachment to a proper specific sequence, identification of place, getting inserted into it, and this whole sequence, you can imagine the automaticness of this. Then preparedness of the DNA to get cloned, whether DNA allow itself to get cloned, creation of recombinant DNA, introduction of recombinant DNA to the host organism, selection of organisms they are containing recombinant DNA, then screening for clones with desired DNA insert and biological properties. So these are various activities which can be there, which can be seen into it. Then next is DNA expression, how the DNA is expressed. Then what should be the properties of various organisms which are containing recombinant DNAs. So in most cases, organisms containing recombinant DNA have apparently normal phenotypes, means their phenotypes, their functions are similar, even though they are having Their appearance, behavior, metabolism are usually unchanged, and the only way to demonstrate the presence of recombinant sequence is to examine their DNA by using polymerase chain or RT-PCRs. There are some exceptions are there, and accordingly we can find that they are present. We can check it using RT-PCR, that is reverse transcripted PCR. This is reverse transcription PCR will be showing any such changes are there, any changes in the phenotype, they will be there, or Western hybridization technique or Western blotting is there. That means various proteins which are there, the changes in the protein can be seen into it. These are various cases. The recombinant DNA can have deleterious effects even if it is not expressed, because they can be either immunological. Even though it is not expressed, it is not resulting into any protein, still it can be dangerous one, and by which it causes is insertional activation. That is, once they are inserted, they can, or we call it as a gene silencing, if you remember. So in which the recombinant DNA become inserted into host cells gene. They use this phenomenon to cause the knockout gene. If you remember, I told you these are called as a knockout gene. Or there are animals, those who want to study it, we call, you just search it on Google or anywhere, knockout gene, what it is. So some gene is suppressed, or it is not allowed to express, or it is silenced, or it is removed. So this will cause their biological function and importance. There are many such knockout animals are present, and which are used in various research purposes. Many knockout or genetically modified animals are there, which are used for study of various things. I told you example was one was Papa rat, Papa mice, which was obese one. There are many knockout mice are available, which are various mice are there, and because of their knockout for one or other kind of gene, that particular maybe enzyme, that particular protein, that particular receptor will not be available, and then you will able to identify what other activity the that particular drug or anything is having on this knockout animals. Other mechanism by which this recombinant DNA is inserted into chromosomal DNA can affect DNA expression is by inappropriate activation of previously unexpressed host cell genes. Now there are certain genes and they are to be expressed after certain time. Example, we know puberty genes are there which are activated after puberty, and then the male or female gonadotropic hormones are secreted and there are changes. So the genes are present but they were not expressed because there is some timeline. So they can affect this gene expression because of that. And when recombinant DNA fragment containing an active promoter becomes located next to previously silent host cell, or when host cell gene that functions to restrain gene expression, it undergoes insertional inactivation by recombinant DNA. So these are the top problems which are there. Either it can show silencing, it can cause alteration, it can change the periods, etc., and accordingly it will be there. So recombinant DNA is widely used in biotechnology, it is used in medicine, it is used in research, and there are many such recombinant protein, their products are available, many are used in a medicines, they are produced from their organism, they are present into many places, many supermarket, home medications, etc. There are those which sell glow fish, the fish which is having color, then genetically modified animal which are having different shape or different colors than the normal one. So these are various genetically, and in research I told you there are many research, in human research, in animal research there are many animals are there which are being used. So these are various recombinant are there. I told you various recombinant products are available. Example, recombinant chymosin, recombinant human insulin, recombinant growth hormone, recombinant clotting factors, recombinant virus, hepatitis viruses, diagnostic kits for infected with HIV, and then golden rice. Then if you have heard the word BT cotton, that is Balgard cotton. These are various such informations are there. So these are what we call it as a genetic recombination, and these are helping us to go for various kind of newer genetic phenotypes, and this can be used, this can be utilized, and there are many are available as I told you for various purposes. Rather, this has opened a very big area in medicines and medical research, particularly because of changes in the animals, research animals, and availability of insulin-like molecules or many drugs. So I hope this much is okay. You have understood this, and if you have any difficulties, you may please write it,